Good afternoon, everyone. Today's topic will be histology of liver. Now, in histology of liver, as you can see that it is an accessory gland of gastrointestinal system. Okay. In GIT, last few weeks we have studied about the histology of GIT, where you have seen one thing was common. What is that? Yes. One thing was common. What was that in GIT? Yes, very good. Four basic layers. If you remember, we have started from esophagus, okay, and then we have ended up to anal canal. In that, you have seen one thing was common. That was four basic layers, but some modification was there. Some modifications was there on that four basic layer, but four basic layer was same. Some changes we have seen in case of gallbladder. If you remember, some changes we have seen in case of gallbladder last week. We have discussed about that. Okay. Now, in case of liver, everything will change. So those four basic layers will not be present. Okay. Instead of that, a new concept will be discussed today. Okay. So from now onwards, as we will go forward in the other parts of systemic histology, so mostly we'll see that the gland or the organ or the structure which is present, around that there will be a connective tissue capsule. That connective tissue capsule will be sending septa or trabeculae into the gland, and that will divide it into small lobules or small spaces or small compartments. I hope I have explained you this concept in case of lymphatic system. The organs which you have studied in case of lymphatic system. Lymph node, spleen, then thymus. Okay? That thing was discussed that time. So the same thing will be repeated over here. Next, another important point which you have to add, which you have to know over here is the stroma and parenchyma. Okay? So what is the meaning of stroma and what is the meaning of parenchyma? Stroma means the supporting part. S for stroma, S for supporting. Okay? So stroma means the supporting part of the gland. For example, that connective tissue capsule, blood vessels, nerves, connective tissue septa, lymphatics, then reticular fibers forming the network framework. Okay, in case of liver, those will come under supporting part. That's why they are called as stroma. Next, what is parenchyma? Parenchyma means the producing part or the secreting part. In this case, the parenchyma will be the hepatocytes. So we know that in the liver, hepatocytes cells are present, and the main function of those hepatocytes are to produce bile. Okay, so that is production or secretion, and that is done by the parenchyma. Okay, so these are two aspects of the glands or the structures which we are going to study from here. Same thing will be applied in case of pancreas also. Okay, so these are the two things. Next, another important point or another important part which you have to see over here is the exocrine part of the liver. Okay, now what is this exocrine part of the liver? Inside the liver, intrahepatic biliary system is present. Liver से जो bile secrete होता है, everyone you know that from liver bile is secreted. After secretion of the bile, where does it goes? Okay, gallbladder, very good. And after gallbladder, where it will go? Which part of the intestine? Yes, duodenum. And you know the function also. Okay. Now, once it comes out from the liver and it will go and store inside the gallbladder for concentration, that part is called as extrahepatic biliary system. Extrahepatic. Extrahepatic means outside the liver. Okay. Now I am talking about the intrahepatic part. So what is the meaning of intrahepatic? That means the biliary system which is conducting the bile straight away from the hepatocytes. Okay. The small canaliculi, the small ductules which are present. That is the intrahepatic part of the biliary system. That is also included over here. So as a whole, so many things are there which you have to consider and you have to study. That's why the microscopic structure of liver is quite complicated. It's not that simple. Identification is very simple. I'll show you the slide. Okay, if you see it once, you can identify, no problem. But when you will try to understand the concept, when you will try to write it in the exam or in the practical notebook, at that time you will see it's quite difficult. It's not that simple. Okay, already I told you what are the things you have to consider. Okay. So let's start. So liver, as you know, it is an accessory gland of GIT. Okay, and it is made up of two components. What are these two components? Stroma and parenchyma. So what is stroma? What is parenchyma? I have explained. Next, if you see the outer surface of the liver, first of all, it is covered by peritoneum. Okay, liver is a intraperitoneal organ. Next month onwards, we will start with the abdomen. Okay, at that time we will come to know peritoneum, peritoneal cavity, peritoneal folds, etc. So liver is an intraperitoneal organ. There are some areas of the liver which are not covered by peritoneum, called as bare areas of the liver. Okay, but mostly it is covered by peritoneum. So first of all, a peritoneal layer will be there, serous peritoneum. Now deep to that, another layer of connective tissue is present, and that is called as Glisson's capsule. Okay, in case of liver, it is called as Glisson's capsule. Now beneath the serous coat, a thin layer of connective tissue is there, that is called as Glisson's capsule. Now this Glisson's capsule will entirely cover the liver. Okay. Next, from there, it extends into the interior as numerous branching septa and trabeculae. तो इसका मतलब क्या है? From this Glisson's capsule, numerous branching septa and trabeculae will extend into the gland, or will extend inside the liver, or you can say extend into the parenchyma. After that, what will happen? The radicals of the portal vein, hepatic artery, and bile ductules run together with this septa. तो ये जो septa जाता है, along with that septa, it is carrying these structures, portal vein, radicals of the portal vein, hepatic arterioles, bile ductules, etc. These are also passing through this trabeculae or septa. Okay. Next, we have to see separately what is there in the stroma and separately what is there in the parenchyma. Okay. So in the stroma, you are getting the supporting part. First of all, we are getting the Glisson's capsule, an extension from the Glisson's capsule, which are in the form of trabeculae and septa. Okay. Along with that, what we are getting? Lymphatic vessels, bile ducts, okay, nerves which travel along with the connective tissue. So that is the part of stroma. Next, this connective tissue septa which is extending, it will branch extensively, okay, inside the parenchyma of the liver, and it will divide it into small structural units, and those are called as liver lobule. I'll give you an example. Okay, just imagine this is a classroom and it is having certain boundaries. This is one wall, that is one wall. In between that, if I make some 2 or 3 more walls, then what will happen? Suppose ये क्लासरूम है, ठीक है, इसके अंदर मैंने यहाँ पे एक वॉल दिया है, यहाँ पे एक वॉल दिया, तो क्या होगा? पार्टीशन हो जाएगा, स्मॉल कंपार्टमेंट में डिवाइड हो जाएगा. ठीक है, तो सिमिलरली क्या होता है, लिवर के बाहर जो ग्लिसन्स कैप्सूल रहता है, इट इज सेंडिंग सेप्टा और यू कैन से पार्टीशन that is extending inside the liver, and it is dividing into small, or you can say classical liver lobule. So the parenchyma of the liver by those septa is divided into small lobules, and those are called as classical liver lobule, and these are the basic structural unit of the liver. I hope that much is clear. Next, again in the supporting role, we are getting the reticular fibers, which are nothing but the type 3 collagen fibers, and they are in the form of reticulum, okay, which is supporting the stroma of the liver. Okay, so you can see reticular fibers, they are finer reticular fibers in the form of network, and they supports the parenchyma of the liver. Okay, so that will also come under the stroma of the liver. Okay, the supporting part. So in the supporting part, what are the things we have studied so far? First of all, Glisson's capsule. From Glisson's capsule, we are getting connective tissue, septa and trabeculae. After that, with that septa and trabeculae, blood vessels, nerves, lymphatics, all are traveling. Okay, so those will also come as a supporting role. Next, what we are getting? we are getting this connective tissue septa is dividing the liver into small structural units, and those are called as classical liver lobule. Now this is very important. When you will see the slide of the liver under low magnification, these are the things which you have to see first. Okay, and that can be seen very easily under low magnification. Next, in the supporting role, what we are getting? Reticular fibers. Okay, those are nothing but the type 3 collagen fibers, and they are in the form of reticulum or network, which is also supporting the parenchyma of the liver. Okay, so that overall constitutes the stroma of the liver. Next is the parenchyma. The parenchyma में क्या है? Mainly we are getting the hepatocytes. Okay, so in case of parenchyma, it consists hepatocytes that also they are highly organized in case of liver. Okay. Now, which in case of adult, they are one cell thick and are separated from each other by sinusoidal capillaries. इसका मतलब क्या है? First of all, the parenchyma is made up of what? Parenchyma is made up of hepatocytes. Now, hepatocytes are highly organized in liver. Okay. Now, the way they are organized, okay, it is forming a layer of cells, and that layer of cells, so many cells are there in one layer, but the layer is made up of only one cell. That's why it is called one cell thick. ठीक है, multiple layers cells नहीं रहता है, एक ही layer में cells रहेगा. Okay, and that is in the form of a layer, and it is also called as laminae. ठीक है, तो सपोज एक लेयर है और एक लेयर है, दोनों लेयर के बीच में एक छोटा सा गैप है, and that is filled by, or you can say that is separated by sinusoidal capillaries. So two layers or two laminae are separated from each other by sinusoidal capillaries. Now for a reference, you can see this diagram. The first of all, you can see this is the interlobular septum, which is coming from the Glisson's capsule. After that, this is the arrangement of hepatocytes. You can see these are the arrangements of hepatocytes. Okay, spatial or you can say definite arrangements are there, and that also in the form of laminae. Now adjacent laminae are separated from each other by, you can see these are the sinusoids. Okay. Now in the sinusoids, if you see endothelial cells are there, along with the endothelial cells, specialized cells are present, and those are called as Kupffer cells. Okay. So I am coming into this. What are Kupffer cells? What are the functions of Kupffer cells? So this is the organization of which part of the liver? This is the parenchyma of the liver. Okay, and this particular part, what you see over here, this is the stroma part, and this is the parenchyma of the liver. Okay. Next, hepatic sinusoids are also there. So here I have shown you the hepatic sinusoids. Here you can see these are the hepatic sinusoids. Okay, so in the hepatic sinusoids, what happened? These are fenestrated sinusoids where endothelial cells and Kupffer cells are present. Okay. Now, blood from the sinusoids open into the central vein that occupies the center of the lobule. So, blood from the sinusoids, they will go and they will open into the central vein. So, one central vein is present over here. So, in that the blood is draining. That is the direction of the flow of the blood. Please keep that in mind. In case of liver, the flow of blood and the flow of bile is opposite to each other. Okay, flow of blood and flow of bile is opposite to each other. Okay, next, if we go deep into the parenchyma, then you can notice some other points also. There is the substance of the liver. Okay, divisible into large lobes which can consist numerous lobules. So first of all, that connective tissue septa will divide into lobes. After that, what we can do, it consists of numerous lobules. Now the parenchyma of the liver is divided into units which are based in three ways. So liver lobules are there. Connective tissue septa will divide into liver lobules. Now those liver lobules, okay, are divided into different units. And they are classical liver lobule, portal lobule and liver acinus. Further division. ठीक है, तो पहले क्या होता है? The connective tissue septa is dividing into lobules, and from those liver lobules, what we are getting further, it is divided, okay, into classical liver lobule, then portal lobule, and then liver acinus. So we have to understand this. Now this will constitute the main parenchyma of the liver. Okay. Now if you see clearly here, the classical liver lobule, the shape is hexagonal shaped. Next, in case of portal lobule, it is triangular shaped, and in case of liver acinus, it is elliptical shape or also in diamond shape. Okay. Next, here in the center, we are getting the central vein. Here in the center, we are getting the portal triad. Now you have to understand what exactly is this portal triad and what are the structures present in the portal triad. Okay. And in case of liver acinus, two axes are there, short axis and long axis. And in between them, we are getting three different zones. Okay. So let's start with the classical liver lobule. So what is the shape of the classical liver lobule? Hexagonal shape. I hope everyone you know what is hexagonal shape. So what is the shape of this part? Is it hexagonal shape? Okay, so it is hexagonal in shape. So classical liver lobule, first of all, it is hexagonal in shape, and at the center you are getting a central vein. Okay. Now from the central vein you can see the hepatocytes are radiating towards the periphery. As you can see from the center, the hepatocytes are radiating towards the periphery. Now if you go towards the periphery, at the angle of each part of the hexagon, you can see a structure, and that is called as portal triad. So this is the hexagonal shape. This is the center. From the center, if you go towards the periphery, that means near the angles of the hexagon, then what is visible? You can see these structures. These are called as portal triad. Okay. So why triad? Because three things are present in this particular area. Now this particular area, what happens? Actually, it is covered by connective tissue layer, and it is converting this particular area into a canal, and it is called as portal canal. Inside the portal canal, three important structures are present. Okay, and that's why it is called as portal triad. Next, another important part which you can observe over here, from the central vein, the hepatocytes are radiating towards the periphery, and in between the laminae of the hepatocytes, the spaces or the area which you can see, these are the sinusoids. Okay. तो इसका मतलब क्या है? ये central vein है, ठीक है? Now this is one layer of hepatocyte. This is one layer of hepatocytes. In between this, you can see this particular area. This is the sinusoids. ठीक है? तो क्या होता है? The blood from this sinusoids is draining towards the central vein. So that is the direction of flow of the blood. If this is direction of flow of the blood, what is the direction of flow of bile? Opposite. So it will go towards the periphery. ठीक है? So that is the direction of flow of bile. So this is the classical liver lobule. Now, as I have mentioned, at the angle of the hexagon, portal triads are there. Okay, and these portal triads are running in the portal canal, which is the connective tissue area inside the liver. Now, one important space is present over here, and that is called as space of Moll. M-A-L-L, Moll, space of Moll. Now, what is this space of Moll? Where it is present exactly, and what is the function? Now, this is a 3D diagram where you can see the space of Moll. So, this is the central vein, and which area is this? This is the portal triad, and you can see the structures inside the portal triad. Okay. So, space of Moll is a space between the connective tissue stroma of the portal canal and outermost hepatocyte. So, these particular structures are surrounded by a layer of connective tissue. Okay. And outer to that, you can see a layer of hepatocytes are present. In between these two structure is potential space which is present. It is called as space of Moll. Okay. Lymphatics of the liver starts from the space of Moll. Okay, lymphatics of the liver starts from the space of Moll. Now, in case of lower four-footed animals, for example, pig, classical lobules are clearly seen. Okay, classical liver lobules are clearly seen. That means you can differentiate between the classical liver lobules. Why? Because thick connective tissue. Because the connective tissue which is present, that is very thick. and it can be differentiated very well. The classical liver lobules can be differentiated very well. But in case of human being, that connective tissue part which is present, that is not significant, or you can say that is not prominent. That's why when you will see the slide of liver under microscope, it will be very difficult to differentiate between the individual classical liver lobule. Okay, liver lobule है वो दिखाई देगा, यहाँ पे एक है, यहाँ पे एक है, like this you can't differentiate. Why? Because the connective tissue which is present around the classical liver lobule, it is not prominent. In case of lower animals, these are prominent. Okay. Next is the portal lobule. So what is this portal lobule? You can see this is the portal lobule and this is the portal canal. So I was talking about this portal canal through which the structures are passing. Okay. Now, what is this portal lobule? It is a triangular area of the liver parenchyma. As you can see, this is the triangular area of liver parenchyma, which is formed by joining the central veins of the classical liver lobule. Okay, so these are the central veins of the classical liver lobule. If you join them, then you will be getting the portal lobule. Okay, now in the portal lobule, it is considered as functional lobule as the bile duct lies at the center. तो यहाँ पे पोर्टल ट्रायड है, पोर्टल ट्रायड में जो बाइल डक्ट होता है that lies at the center, okay, of the portal lobule. Next, portal canal. Already I have told you what is this portal canal. Now in the portal canal, what are the constituents present in the portal canal? As you can see in the diagram, see this is one portal canal, this is one portal canal. Okay, radicle of the portal vein, hepatic arteriole and interlobular bile duct. Okay, so what are the structures present? Radicle of the portal vein, hepatic arteriole and interlobular bile duct. These are the structures, three structures present inside the portal canal. Now blood from the branch of portal vein and the branch of hepatic artery enters the sinusoid directly, okay, at the periphery of the lobule and pass towards the center. Now what does it mean? If you see this diagram, what you can see in this diagram? So this particular part, okay, is the portal canal, and inside the portal canal you can see three structures are present. That's why it is called as portal triad. Now what are those three structures? First you are getting the hepatic arteriole, then a portal venule and interlobular bile duct. Okay, these are the three structures. Now if you consider the portal venule, okay, radicle of the portal venule and the hepatic arteriole, can you notice anything? क्या दिखाई दे रहा है इसमें? तो दिस इज द वेन, राइट? तो यहाँ से जो ब्लड आ रहा है एंड दिस इज द आर्टेरियल मींस आर्टरी, यहाँ से जो ब्लड आ रहा है, बोथ आर मिक्सिंग टुगेदर इन द साइनोसॉइड. So liver is the only organ in our body where the mixing of blood is taking place, as you can see in the diagram. Okay, and then it will pass through the sinusoid and then it will go to the central vein. Now can anyone tell me why the mixing of blood is taking place? In case of hepatic arteriole, the blood which is coming, ठीक है, that is oxygen rich blood. तो oxygen rich blood is required for the hepatocytes. Normal जो cells है उसको oxygen चाहिए. Next, what about the portal vein? Now here the venous blood means it is nutrient rich blood. ठीक है, nutrient rich blood. तो अभी सबने लंच किया होगा, सबने लंच किया, हाँ. तो after digestion for metabolism, where it goes to the liver. How it will go to the liver? Through this portal vein. So whatever you have eaten after digestion, when absorption will take place, at that time the venous blood that last week in the intestine chapter I have shown you the structure of villi. At that time I have mentioned this particular point. ठीक है, तो after absorption through venous blood, where it will go to the liver. How it will go by portal vein. तो portal vein में जो blood रहता है, that is nutrient rich blood, because it is mainly coming from the gut. ठीक है, so that's why the mixing is taking place. Further, I know you have studied in physiology. ठीक है, इससे ज़्यादा if I go, it will take 1 hour or 1.5 hours. I can show you the slide. Physiology, have you studied all those things in physiology? I know you have studied all those things. Recently or a few months ago? When you have studied? Recently. So see, recently you have studied, but you don't know all those things. Anyway, so these are the structures of the portal triad. Okay. Next is the portal acinus. So what is the shape of the portal acinus? It is diamond shaped. As you can see, portal acinus, it is diamond shaped area. This one, ye wala. Can you see the portal acinus? Yes. There is a diamond shaped smaller unit of the liver tissue, and it is supplied by one individual hepatic arteriole. Now, if you see the hepatic arteriole, it is running along the junction of the two hepatic lobules. So, two hepatic lobules are there. In between the junction of the two hepatic lobules, this particular hepatic arteriole is running, and it is supplying the portal acinus. Now, the central veins are situated at the ends of the acinus. They are also called as the functional unit of liver. Okay. Now, here you are getting two axes, one long axis and one short axis. Now, if you see the short axis, here this is the short axis. Now, the short axis is in between the two portal triad. Okay. And the long axis between the two closest central vein. So here you will be getting one portal triad, here you will be getting one portal triad. If you join these two portal triad, then you are getting the short axis. And if you join the two central vein adjacent or nearest to central vein, then we are getting the long axis. Okay? Now, in between these two axes, three zones are present. As you can see in the diagram, zone 1, zone 2 and zone 3. तो क्या होता है एग्जैक्टली तो यहाँ से हेपेटिक आर्टेरियोल आएगा एंड देन इट विल सप्लाई द एसिनस. तो zone 1 will have rich blood supply, zone 2 is intermediate and zone 3 is having less blood supply. So the area which is having less blood supply, it is more prone to hypoxic injuries. Okay, that is the significance of this particular zones, zone 1, 2 and 3. Okay, so that is the portal acinus. Next, if you see the hepatocytes which are arranged, okay, as I have mentioned, hepatocytes are in the form of laminae. Okay, now they are separated from each other by hepatic sinusoids. Now, if you see the sinusoids, exactly what is present in the sinusoids? So, this is the sinusoids. Okay, these are the sinusoids. Now, in case of sinusoids, two important cells are present. First of all, if you see the lining epithelium, that is the endothelium, they are separated from each other. Generally, जो endothelium रहता है, in case of blood vessels, especially arteries, vein, they are continuous with each other. But here, it is separated from each other, as you can see in the diagram. ये देखो, gap है, ठीक है? Next, इसका जो basal lamina है, in that also gaps are present. Okay, so that is the speciality of the sinusoids which are present in case of liver. Next, stellate sinusoidal cells or Kupffer cells are present. Okay, so Kupffer cells are present. Now, what is the function of these Kupffer cells? Phagocytic activities. Okay, they are having phagocytic activities. Now, a very important space is related to this particular area, and that is called as space of Dis. Previously, we have seen space of Moll. Now, around the sinusoids, a space is there that is called as space of Dis, D-I-S-S-E. Okay? Now, that is also called as perisinusoidal space. Now, how it is formed? So, sinusoidal epithelium separated by the underlying hepatocytes, okay, by a small extracellular space. So, if you notice over here, see, this is the space of Dis. तो यहाँ पे the endothelial cells which are present and the hepatocytes which are present, in between them you can see a small space is there, and that is called as space of Dis. ठीक है? तो जो sinusoids होता है, adjacent to the sinusoids, the hepatocytes are present. So in between them a small space is there, and that is called as space of Dis. Now the question is, what is the importance of the space of Dis and what is present in the space of Dis? Okay, so you can see the space of Dis is the site for formation of blood cells, especially RBCs in case of fetal life. You have studied that in fetal life, liver is acting as hemopoietic organ. That means the formation of the blood cells are taking place in the liver during intrauterine life. Okay, so that is forming in this particular area. Next, the hepatic stellate cell or ETO cells are there, okay, which are present in this particular area. Now, these ETO cells are involved in storage of vitamin A and lipids. Okay, so these are some important functions of the space of DIS. Next, another important function is there. For that, I will go back to the previous diagram. हाँ. Now, if you see this particular diagram, यहाँ पे भी दिखाई दे रहा है. See, this is the space of DIS. Now, what happens? यहाँ पे जो hepatocytes है, when I will come into the EM structure of hepatocyte, at that time you will come to know that hepatocyte का तीन surface होता है. Three different surfaces are there of the hepatocytes. ठीक है? So the surface of the hepatocytes which are towards the sinusoid, उसमें क्या होता है? Microvilli रहता है. So what will happen? From this hepatocyte, the microvilli will extend towards the space of DIS. Now, why it is required? Microvilli का function क्या है? हाँ, absorption तो है, surface area increase करेगा to absorb. Now, what is absorb? तो through this the blood is going, nutrient rich blood, oxygen rich blood is going, तो जो nutrient absorb होगा, okay, for that this microvilli are present. Okay, so that is the function and importance of the space of DIS. Okay, next is the exocrine part of the liver. In the exocrine part of the liver, first of all, you have to study about the intrahepatic biliary system. Okay, so can you see these small ducts? Can you see these small ducts? Wait, let me go to the diagram again. So if you see this particular diagram, this 3D diagram, you can understand it in a proper way. तो क्या होता है? Bile जो produce होता है liver में, okay, that constitute the exocrine part because bile is not drained into the blood directly. Bile blood में नहीं आता है, ठीक है? It should not come to the blood. Where it has to go? It has to go to the second part of the duodenum. Now, this pathway which is followed by the bile, starting from the production to the second part of the duodenum, it is divided into two parts, intrahepatic part and extrahepatic part. तो यहाँ से intrahepatic part start होता है. Okay, so you can see these are the hepatocytes. So hepatocytes, what it will do? It will produce the bile. Once it produces the bile, first it will go to the bile ductules, or you can say bile canaliculi. From bile canaliculi, it will go to the bile ductules. Then intralobular ductules, interlobular ductules. Then they will join together to form the right and left hepatic duct. Those will join together to form the common hepatic duct. So like this, the duct system is formed. And lastly, we are getting the common bile duct when it is joined by the cystic duct. Okay? So like this, the formation is taking place. So you can see the formation of bile. After that, what will be the direction? तो यहाँ से form होगा. This will be the flow of the bile. So as I mentioned, flow of the bile and flow of the blood is opposite to each other. So how the blood is flowing? This side. So how the bile will flow? This side. ठीक है? तो यहाँ से production होगा and then it will come towards the bile duct. Okay? So this is the how bile is flowing. Now for that, what you have to understand, that is the intrahepatic biliary system or intrahepatic biliary tree. So this diagram is showing the intrahepatic biliary tree. As we come from the bile canaliculi towards the common hepatic duct, you can see the diameter increases. जितना बड़ा duct होगा, उसका diameter increase होगा as usual. So intrahepatic biliary tree drains the exocrine part of the liver, that is the bile, and conveys the extrahepatic biliary tree through right and left hepatic duct. Okay. First of all, we are getting the bile canaliculi. So canaliculi are the spaces between the plasma membrane of adjacent hepatocyte. तो one hepatocyte, another hepatocyte, दोनों का plasma membrane के बीच में जो space है, that is nothing but the bile canaliculi. So once the bile is produced, directly it will be poured into the bile canaliculi. ठीक है? Next, from bile canaliculi, we are getting the intralobular ductules, or also called as canal of Herring, which I have shown you just now. So bile canaliculi drain the bile into intralobular ductules. Now these intralobular ductules are lined by simple cuboidal epithelium. Okay, intralobular ductules are lined by simple cuboidal epithelium. From there, we are getting the interlobular duct. Okay, so this from intra, we are getting the interlobular ductules. Now, these interlobular ductules are lined by a simple cuboidal to columnar epithelium. Okay. After that, we are getting the right and left hepatic ducts. So, this interlobular ducts will join to form the right and left hepatic ducts. After that, it will come out from the liver. Okay. After that, the extrahepatic biliary part starts. Okay. So, this is the exocrine part of the liver. Now, here I have described, or I have explained up to the intrahepatic part only. Extrahepatic part not required right now, as we'll start the abdomen. At that time we'll discuss about the extrahepatic part. There is a very important topic in the abdomen called extrahepatic biliary system. Okay, extrahepatic biliary system, very important. Okay, because I have seen so many times questions are being asked from this particular topic. Okay, so that includes the extrahepatic parts, starting from here. Now this is the direction through which the bile is flowing, as you can see in the diagram. तो पहले कहाँ से production होता है bile का? Hepatocytes. After that, where it goes, it is going to the bile canaliculi. From bile canaliculi, it is going to the canal of Herring, or also called as intrahepatic ductules. From intrahepatic ductules, it is going to the intralobular bile ducts. This is a part of portal triad. Now here you can see these are lined by simple cuboidal to columnar epithelium. Okay, these are lined by simple cuboidal. Here both cuboidal as well as columnar epithelium is there, and after that right and left hepatic ducts followed by the common hepatic duct. Okay, so common hepatic duct means it has came out, or it has come out from the liver. So this is the direction of the flow of bile. Next, structure of hepatocytes. So several times I have mentioned that bile is produced by the hepatocyte. Now if you want to see the hepatocytes, what you have to do? You have to use EM structure. Under normal microscope, it is very difficult to see the hepatocytes. Okay? So this is the diagram showing the EM structure of hepatocyte. तो क्या दिखाई दे रहा है? What you can see over here? First of all, you know that the liver parenchyma is mainly made up of hepatocytes. What is the shape of the hepatocyte? Polygonal shaped, large polygonal shaped. If you see the dimension, 20 to 30 micrometer in size. Okay, if you see the nucleus, single nucleus. Sometimes it is binucleated also, but mostly it is single nucleus, large, spherical, and it is at the center. Now, nucleus is highly active, having huge euchromatin. Okay, and the lifespan is approximately 5 months, and it is having high regenerative capacity. Okay, agar destroy hua, easily it can be regenerated. And cytoplasm is highly acidophilic in nature. So, if it is acidophilic, what will be the appearance? Highly acidophilic, what will be the appearance? Eosinophilic, pink appearance hoga. Next, in the next diagram, you can see the surfaces of the hepatocytes. How many surfaces are there? 3 surfaces are there. Okay, sinusoidal surface, canalicular surface, and intercellular surface. Sinusoidal surface means the surface of the liver which faces towards the hepatic sinusoid. Okay, so you can see this is the hepatic sinusoid. This is the sinusoidal surface. तो यहाँ पे space of disc रहता है, ठीक है, and you can see the microvilli are extending from the sinusoidal surface towards the space of disc, and we have discussed what is the function in this case. Next, canalicular surface. Now, what is the canalicular surface? It forms the wall of the bile canalicus, and if you see the wall of the bile canalicus, it is approximately 0.5 micrometer. Okay, so adjacent. So this is one hepatocyte. This is one hepatocyte. Okay. Now, in the canalicular surface, you can see bile canaliculi are present. Okay. Now, the bile canaliculi which are present, as I said, जो दो hepatocytes है, उसका जो plasma membrane है, उसके बीच में जो gap है, that is forming the bile canaliculus, and that is the smallest unit of the exocrine part of the liver. Okay. Now, once the bile is produced, it is directly poured into the bile canaliculus. It will come directly over here. Okay. So that is the canalicular surface. And another surface is that is called intercellular surface that follows towards the adjacent hepatocyte. So this is one hepatocyte. This is another hepatocyte, which is facing one opposite to another. That is the intercellular surface. Okay, so these are the three different surfaces of the hepatocytes. So that is all about the histology of liver. Now, if I ask you what are the different types of cells we have studied in liver, can you recall? First are the hepatocytes. What is the location? Or you can see cords or plates. You can see the main function. Then we got the cover cells lining the sinusoids, main function phagocytosis. Ito cells are there, or also called as hepatic stellate cell. Perisinusoidal space of this. What is the function? Storage of vitamin A and lipid. Then sinusoidal endothelium, which is present in the sinusoids. Okay, and hemopoietic cells in the perisinusoidal space of this form, and that is in the intrauterine life. Okay. So till now I have not shown you the slide of liver. We have explained, we have discussed everything related to the liver. Now this is the slide, or you can say schematic diagram of the histology of liver under low magnification. तो क्या दिखाई दे रहा है? So first what you have to see the classical liver lobules, which are hexagonal shape. So this is one classical liver lobule. This is one classical liver lobule. This is one classical liver lobule. Okay, which are hexagonal in shape. Next, what you have to see, you have to focus on one particular classical liver lobule. See at the center, what you will get? Central vein. And from the central vein, the hepatocytes are radiating towards the periphery. Okay? In between the laminae of the hepatocytes, you can see small empty spaces. These are nothing but the hepatic sinusoids. Okay? Now, it's not possible to see the covered cells, okay, because uske liye alag sa different type of stains are required. Anyway, if you go towards the periphery, you will be getting the portal triad. See, this is one portal triad, this is one portal triad. So you will be getting the portal triad. So try to focus on the portal triad. You will be getting these structures. Now the question is how to identify these structures in the portal triad. Three things are there in the portal triad. What are they? First is the portal venule, radicle of the portal venule, then hepatic arteriole and interlobular bile ductule. So the best way to identify is जो जिसका size सबसे बड़ा होगा, जिसका diameter सबसे बड़ा होगा, that is the portal venule, radicle of the portal venule, उसका size सबसे बड़ा होता है, ठीक है. Next, in between these two structure, please note that जो intralobular ductule होता है, bile ductule होता है, that is mainly lined by simple cuboidal epithelium, cuboidal to columnar epithelium. But जो arteriole होगा, that will be lined by simple squamous epithelium, that is the endothelium. So this is how it can be differentiated. Okay. So you can see the portal venule, the large one. After that, you can see the bile ductule. Now, if you concentrate over here, can you see the simple cuboidal epithelium? Yes. But here you will see the simple squamous epithelium or the endothelium. So this is the way to differentiate. Now, can you tell me what is the difference in this image and in this image? This is the schematic diagram. Okay? Here, can you see the classical liver lobules? Clearly, you can differentiate. Can you differentiate the classical liver lobule? Why? Because in case of human being, the connective tissue septa which are present, they are not prominent in nature. That's why in the slide, it cannot differentiate between the individual classical liver lobule, but they are present. Okay? Apart from this, you can identify what is this. This is the central vein. Okay? यहाँ पे भी एक central vein है. Next, you can see the portal triad. You can see the cords of the hepatocytes. Now, if you see under high magnification, then here you can see the portal triad. ठीक है? तो इसमें क्या दिखाई दे रहा है? You can see the portal vein. You can see the hepatic arteriole and you can see the bile ductule. ये देखो bile ductule दिखाई दे रहा है. तो इसमें just concentrate on the lining epithelium and concentrate on the lining epithelium over here. You can see the differences. Okay. Next, so this is the portal triad. Then hepatic sinusoids can be seen. ये जो empty spaces है, these are hepatic sinusoid, and you can see the hepatocytes also. Okay. So that's all about the histology of liver.